This is an overview of an article titled Genetic Reduction in Left Ventricular Protein Kinase C Alpha and Adverse Ventricular Remodeling in Human Subjects, published in the March 2018 issue of Circulation, Genomic and Precision Medicine. Protein kinase C alpha is a serine threonine kinase operating downstream of signal transduction pathways to help regulate myocardial function. Either activation or increased expression of PKCα is known to be associated with cardiac hypertrophy, cardiomyopathy, ischemic injury, or neuroendocrine stimulation. In contrast, PKCα inhibition in animal models enhances contractility and cardioprotection, which therefore has been proposed as a therapeutic strategy for myocardial disease in humans. Although PKCα levels are elevated in failing human myocardium, the impact of PKCα inhibition on human cardiac function remains unknown. The authors of the paper under discussion and others previously demonstrated that common variants within the gene encoding PKCα, known as PRKCA, are linked to reduced gene expression and lower PKCα transcript levels in the human left ventricle, or LV. In this paper, the authors analyzed these variants in detail with the rationale that they might yield an understanding of mechanisms and clinical impact of inherited reduction in ventricular PKCα. Expression quantitative trait loci, or eQTLs, are distinct genomic loci that contribute to messenger RNA expression level variation. Those that act locally, at or close to their gene of origin, are referred to as local or cis-eQTLs, whereas those associating with expression changes occurring distally from their gene of origin are known as distant or trans-eQTLs. The common human genetic variant under examination by the authors of this paper, RS9912468, strongly associates with PKCα transcript abundance in the human left ventricle, and since the variant resides within an intron of the PRKCA gene, RS9912468 can be considered as marking a cis-eQTL for PRKCA. The single nucleotide polymorphism, or SNP, called RS9912468, involves substitution of guanine, or G, in place of the usual cytosine, or C, at a genomic location within the second intron of PRKCA, with C being the major allele and G being the minor allele, that is, the lower frequency allele. The presence of the minor allele is associated with reduced PRKCA messenger RNA expression in both non-failing and failing human hearts, as shown in the left upper panel of the figure shown here. In the right upper panel, Western blotting of human LV tissue protein extracts exhibits reduced PKCα protein expression in minor allele homozygotes, that is, having a GG genotype, versus major allele homozygotes having a CC genotype. The lower panels, from left to right, demonstrate overall protein abundance quantitation by genotype, the high correlation between protein and messenger RNA expression, and the author's finding that when surveying eQTL data from the Gene Tissue Expression, or GTEx, project across 16 human tissues, only left ventricular tissue had a significant eQTL with RS9912468. To understand whether a characteristic feature within the intronic region of PRKCA might account for this LV-specific expression association, the authors examined this eQTL by fine mapping, using 1,000 genomes imputation to test associations between LV PRKCA expression and genotypes near RS9912468. They found that variant RS9912468 marks a single linkage disequilibrium block of about 20 kilobases in length with an intron 2 of PRKCA containing additional variants that strongly associate with PRKCA transcript levels. Within this block, they then carried out chromatin immunoprecipitation sequencing, or ChIP-seq, to look for possible protein DNA associations within this region. They detected genomic sequences exhibiting increased binding of two distinct post-translational modifications of the DNA-binding protein histone 3, specifically acetylation of lysine residue 27, known as H3K27AC, and methylation of lysine residue 4, known as H3K4ME1. H3K27AC is indicative of increased activation of transcription and can represent poised enhancers when subtracted from the H3K4ME1 signature that represents all enhancer regions. On this basis, the authors surmised that this is a putative enhancer region that might be specific for LV tissue. Moreover, two additional common SNPs, RS9910355 and RS9303504, associated with PRKCA expression and physically residing within this putative enhancer, were found by the authors to be in perfect linkage disequilibrium with RS9912468. To test putative enhancer function and specificity in vivo, the authors generated enhanced green fluorescent protein constructs containing the cloned enhancer region, which they microinjected into zebrafish embryos at the single-cell stage. Following injection, detection of fluorescence at 72 hours post-fertilization revealed marked expression in cardiac tissue, as shown in the upper left panel. In the upper right panel, confocal fluorescence imaging of zebrafish hearts demonstrated that this enhancer activity preferentially localized to ventricular tissue over other regions of the heart. To differentially quantify enhancer activity in cardiac and non-cardiac cell types, the authors also cloned the putative enhancer upstream of firefly luciferase and transfected this construct into a non-cardiac line, HeLa, the cardiac-derived HL1 myoblast cell line, and freshly isolated neonatal rat ventricular myocytes. As shown on the lower left, the enhancer region increased luciferase activity, roughly 5-fold only in the cardiac-derived cell lines. The authors thus concluded that in combination, human eQTL data and in vivo and in vitro functional assays support this PRKCA intronic region of interest as a bona fide cardiac-specific enhancer. Because variants RS9910355 and RS9303504 localize within the enhancer, as noted on the previous slide, a full combination of their major and minor alleles together define 4 separate haplotypes. To compare activity of these distinct haplotypes, the authors performed site-directed mutagenesis of the cloned enhancer region to generate all 4 haplotypes, and they compared their ability to drive a luciferase reporter. As shown on the lower right, cells transfected with any of the 4 haplotype enhancer sequences increased luciferase activity relative to controls in neonatal rat ventricular myocytes, but not in HeLa cells. Moreover, haplotypes containing the minor allele G at RS9303504 had luciferase activity 40% lower than the major allele C, whereas expression was unaffected by RS9910355 genotype. Ultimately, these haplotype findings parallel the expected directionality based on the eQTL associations in the human left ventricle. The minor allele G at the lead SNP RS9912468 marks a PRKCA enhancer haplotype containing a minor allele at RS9303504 that reduces enhancer activity and left ventricular PRKCA expression. Having established that this genomic region harbors cardiac-specific enhancer function, the authors focused on the functional impact of the presumptive causal enhancer and gene through expression modulation in zebrafish embryos, and by examining the effect of genotype in human subjects using data from existing genome-wide association studies, or GWASs. In zebrafish, PRKCA knockdown resulted in no observed structural effects on ventricular dimensions. For analysis in humans, clinical referral cohorts were interrogated for measures of LV mass, dilatation, wall thickness, contractile function, all-cause heart failure within the community, and clinical diagnosis of dilated cardiomyopathy. While there were not associations with LV mass by echocardiography or with incident all-cause heart failure, the PKCα reducing allele G at RS9912468 was significantly associated with increased LV mass as measured by electrocardiography, increased LV internal diastolic dimension, LV wall thinning, and a potential decrease in fractional shortening. Furthermore, in a meta-analysis of dilated cardiomyopathy case-control GWAS data, the PKCα reducing allele was associated with dilated cardiomyopathy. Directionality across all of these associations was consistent and suggested that reduced PKCα may be functionally detrimental in humans. This is contradictory to findings in animal models that indicate reduced PKCα expression or activity may instead be cardioprotective. In conclusion, the authors of this study combined genetic, transcriptomic, and epigenetic data to identify a cardiac-specific enhancer region that regulates PRKCA expression in the human left ventricle. Common variants within this enhancer region reduce expression of PRKCA in the human heart, but not in other tissues. And functional studies in vitro and in vivo verify that this enhancer activity is cardiac-specific. Perhaps unexpectedly, as it was contrary to findings in animal models, carriers of the minor allele at RS9912468 that reduces PKCα in the LV display increases in adverse cardiac remodeling and dilated cardiomyopathy risk, which may be dependent upon timing and duration of reduced PKCα abundance or activity. Ultimately, follow-up pharmacologic studies in humans will be required to determine potential benefits and harms of PKCα inhibitors as an approach to treat heart disease.